Once acetyl-CoA enters citric acid cycle with the help of oxaloacetate, which is a 4-carbon compound, it will be converted into a 6-carbon compound citrate by an enzyme citrate synthase. So here is the citrate synthase of chicken, which is shown in figure 14.7 of your book. You can see that this enzyme is a dimer. It is made up of two identical subunits, which are shown here in blue and green. Now this part shows the enzyme without any substrate. However, when substrate like oxaloacetate and acetyl-CoA they bind enzyme, here you can see that a change in the shape of enzyme or a conformation change takes place. Now citrate synthase forms citrate. Once citrate, which is a 6-carbon compound, it is produced, it will be converted into isocitrate. Now isocitrate is an isomer of citrate, and the enzyme involved here is aconitase. So during the conversion of isocitrate from citrate, another intermediate aconitate is also produced. So that's why enzyme involved here is aconitase. Now from isocitrate, the next intermediate of citric acid cycle is a 5-carbon compound, which is alpha-ketoglutarate. During the formation of alpha-ketoglutarate, one of the carbon of isocitrate is released in the form of carbon dioxide, and this step also involves reduction of NAD forming NADH. So now this step, which is catalyzed by enzyme isocitrate dehydrogenase, is known as oxidative decarboxylation, which involves removal of carbon in the form of carbon dioxide. In the next step, from alpha-ketoglutarate, 4-carbon compound succinyl-CoA will be produced, and this step also involves removal of carbon in the form of carbon dioxide along with reduction of NAD forming NADH. Then succinyl-CoA will be converted into succinate, and this step involves formation of energy in the form of GTP in animals. However, in plant cells, it involves formation of ATP from ATP and PI. Now, the remaining steps of citric acid cycle are based upon regeneration of oxaloacetate, as you can see here. That succinyl-CoA will form succinate, succinate will form fumarate, another 4-carbon compound, fumarate will form malate, and malate will be converted into oxaloacetate. Now you can see here that from succinate to fumarate, enzyme is succinate dehydrogenase, which involves reduction of electron transport chain carrier quinone into ubiquinone or reduced ubiquinone. Now fumarate will form malate, and this step involves removal of water. From malate, oxaloacetate will be produced. So if you look at overall equation for citric acid cycle, you will see that acetyl-CoA will be converted into 2 molecules of carbon dioxide plus coenzyme A, and during this process GTP along with 3 molecules of NADH and reduced ubiquinone will be produced. Now, if you look at the bioenergetics of citric acid cycle, if you calculate, you see that at 3 points NADH here, here and here, at 3 points NADH is produced, and I told you that 1 NADH has energy equal to almost 3 molecules of ATP. So 3 here, 3, 6, 9, 10, and this step involves energy equal to 2 molecules of ATP, 12. So it means that when 1 acetyl-CoA enters citric acid cycle, it forms energy equal to 12 molecules of ATP, 3, 6, 9, 10, 12. But if you remember that it is not 1 molecule of acetyl-CoA, in fact 2 molecules of acetyl-CoA from 1 glucose, they initiate or they enter citric acid cycle. So if you multiply 12, if you add 12 plus